Normally, if a human cell accidentally, doubles its entire genetic code, it becomes this bloated, chaotic mess that the body easily flags and destroys.
Right, it just completely falls apart.
Yeah, exactly. But some breast cancer cells actually pull this off, and they become almost completely invisible to the immune system. So today, we are hacking that invisibility cloak.
It is honestly one of the most fascinating paradoxes in oncology right now. The very event that should make a tumor highly visible actually helps it hide.
Okay, let's unpack this. We are looking at a landmark 2026 paper published in Cancer Cell by Foydart and a team out of the Dana-Farber Cancer Institute.
A really incredible team over there.
Yeah, totally. And their mission was to map the exact molecular chain reaction that allows these whole genome doubled or WGD breast cancers to evade immune detection and then, find the specific drugs that exploit that biology to kill them.
And they mapped it all the way from the initial chromosomal error down to the metabolic exhaust driving the evasion.
Which is wild.
It is. To set the baseline here, we really need to look at how whole genome doubling actually happens in a tumor.
Right.
Normally, you have a diploid cell, your standard 2 sets of chromosomes. But in early tumor development, especially when the guardian gene, TP53, is mutated.
The one that usually stops damaged cells from dividing.
Exactly. When that's broken, cells can suffer a catastrophic error. They might undergo cell fusion or experience cytokinesis failure.
Cytokinesis failure. So that's where a cell replicates its DNA to divide, but then just fails to physically split down the middle.
Right. The physical separation just stalls out. And the result is a tetraploid cell, 4 sets of chromosomes.
Right. So instead of 46 chromosomes, you suddenly have 92.
Yep, 92.
And managing that much genetic material has to require immense resources. I mean, think of it like the library analogy. Imagine a perfectly organized library, which is the normal diploid cell.
Nice and tidy.
Yeah, but suddenly a delivery truck dumps an exact duplicate of every single book straight into the aisles.
Real chaos.
Total chaos. It causes massive genomic instability. You'd think the cell would just collapse under its own weight. But the clinical data shows this isn't a death sentence for the cancer at all.
No, in fact, it's the opposite. In highly aggressive subtypes, like triple-negative breast cancer or TNBC and HER2 positive tumors, WGD is incredibly common.
Wow.
And it's heavily correlated with distant metastases and really poor patient outcomes.
The cells shouldn't survive this state, let alone thrive and metastasize.
Right. And to understand why they do, the Dana-Farber team utilized some highly controlled mouse models.
Moving into the lab now.
Yeah. They took mouse mammary tumor cells, specifically the TA3 and 168RN lines, and artificially induced whole genome doubling through cell fusion.
Okay, so they created these matched pairs.
Exactly. The standard diploid tumor cells and then the matched WGD-positive tetraploid versions.
And this is where the experimental design gets really, really elegant. They injected these matched tumor models into 2 totally distinct groups of mice, right?
Yep, 2 groups. First, NSG mice, which are highly immunodeficient. They essentially have no functional immune system.
Like a blank slate.
Right. And second, AJ mice, which are fully immunocompetent, healthy, normal immune systems.
And the growth dynamics between those 2 groups it completely revealed the core mechanism.
It really did. In the immunodeficient mice, the normal tumors and the WGD tumors grew at roughly the same rate.
Okay, so the doubling itself didn't inherently speed up their baseline replication.
Exactly. But in the immunocompetent mice, the ones with an active patrolling immune system, the WGD tumors grew aggressively faster than their normal counterparts.
Wait. If a cell has more DNA, shouldn't it be making more mutant proteins?
You would think so.
Right, shouldn't it be flashing like a neon sign to the immune system? Why is it growing faster when the immune guards are actually on duty?
What's fascinating here is the complete immunological desert they found.
A desert?
Yeah. When they analyzed the tumor microenvironment of those rapidly growing WGD tumors, the CD8 positive T cells,
The assassin cells that actually hunt the cancer.
Exactly, the effector cells. They were essentially absent. And the ratio of those CD8 positive cells to TREGs,
The regulatory T cells, the ones that suppress the immune response.
right, that ratio had plummeted. The WGD tumors were actively maintaining this highly immunosuppressive, cold microenvironment.
So they aren't just ignoring the immune system, they're actively manipulating it. If the CD8 positive T cells aren't even showing up to the tumor bed, that implies a massive signaling failure.
Completely.
Like they must be suppressing the chemokines that normally traffic those cells into the tissue.
That is exactly what the data showed. The researchers looked at chemokine expression and found that WGD cells had virtually shut down the production of CXCL9 and CXCL10.
Wow.
Yeah, these are the critical chemical beacons that call T cells to a site of mutation. Without them, the T cells simply don't even know the tumor exists.
Okay, we need to do a quick recap here for everyone listening, a little bullet summary for segment 1.
Let's do it.
Point 1. WGD-positive tumors grow faster in immunocompetent environments. Point 2, they do this by excluding CD8 positive T cells. And point 3, they create a cold environment by shutting off CXCL9 and CXCL10 beacons.
A perfect summary.
So here's a question for you, the listener, to chew on. Think about your own immune system. If a threat suddenly cuts the phone lines, how do your cellular guards even know where to go?
They don't. But suppressing those chemokines is really only the first layer of the defense.
Because even without a beacon, patrolling immune cells are constantly just sampling the tissue, right?
Exactly. If a T cell bumps into a WGD cell by pure chance, the cancer cell still needs a way to evade recognition.
Which brings us directly to antigen presentation.
Right.
Every nucleated cell in your body uses the MHC-I complex, along with a protein called B2M, to present little peptide fragments on its surface.
It's the cell's molecular ID badge.
Yeah, the ID badge. And if it presents a normal peptide, the immune system just moves on. But if it presents a mutated peptide, the T cell triggers apoptosis and kills it.
And so, if the T cells aren't recognizing the WGD cells even when they bump into them, they must be actively downregulating that MHC-I complex.
They're throwing away their ID badges.
They really are. The WGD cells dramatically reduce their surface expression of MHC-I and B2M. And to prove how vital this specific tactic is, the researchers ran this super fascinating counter-experiment.
Oh, the CRISPR one.
Yes. They used CRISPR to literally knock out the B2M gene in the normal diploid tumor cells.
Which would effectively phenocopy the WGD evasion strategy, right?
Yeah.
By knocking out B2M, they stripped the ID badges off the normal cells.
Exactly. And when they did that, those normal diploid tumors suddenly grew just as rapidly and aggressively as the WGD tumors in the immunocompetent mice.
That is crazy.
They completely isolated the loss of antigen presentation as the definitive driver of their aggressive growth.
But here's where we really need to look at the mechanism.
Mm-hmm.
The WGD cells didn't lose the genetic code for MHC-I or B2M.
No, the genes are still sitting right there in the DNA.
Right. If they aren't mutating the genes away, they have to be silencing them structurally. Are we talking about a widespread epigenetic shift here?
We are. The Dana-Farber team utilized single-cell ATAC-seq for this.
Which is a highly sensitive sequencing method that maps chromatin accessibility for anyone unfamiliar.
Right. It literally shows you which physical regions of the DNA are open and readable by the cell's machinery and which regions are packed away and totally closed off.
And in the WGD cells, the chromatin around those antigen presentation genes was closed.
Tightly closed.
Mm-hmm.
They found a massive global accumulation of a specific repressive epigenetic mark. It's called H3K27E3.
H3K27E3.
Yeah, it involves adding methyl groups to the histone proteins that the DNA wraps around. It forces the chromatin to condense, making the underlying genes physically inaccessible.
It's like molecular duct tape. You're wrapping a critical instruction manual in thick duct tape.
I love that analogy.
Like the information on how to build the MHC-I complex, or how to respond to immune signals like STAT1 and JAK2. The text is still on the page, but the cellular machinery cannot pry the book open to read it.
And this hypermethylation isn't random. If we connect this to the bigger picture, the researchers noted that it coordinates the silencing of the entire interferon gamma response pathway.
Interferon gamma being the primary signal that forces a cell to upregulate its antigen presentation.
Right. By duct taping the chromatin across this specific pathway, the WGD cell makes itself deaf to interferon gamma. It just cannot produce its ID badges.
Okay, let's pause for another quick recap bullet list for you.
Good idea.
Point 1, WGD cells lose their MHC-I ID badges. Point 2, this is caused by closed chromatin and an accumulation of repressive H3K27Me3 marks. And point 3, this acts as molecular duct tape, silencing the entire interferon gamma pathway.
Spot on.
So here's another question for you to think about. If you could chemically dissolve that molecular duct tape, do you think the ID badges would just pop back up?
Well, it is a highly robust defense, but it begs the ultimate question of the whole study. Why does the physical doubling of the chromosomes trigger this specific epigenetic duct tape in the first place.
Exactly. What is the actual bridge between having 92 chromosomes and suddenly overmethylating your histones?
The bridge is cellular metabolism.
Metabolism.
Yeah, and this is where the paper fundamentally shifts how we view cancer biology. Epigenetic marks like H3K27Me3 are regulated by two opposing enzymes.
Okay, a tug of war.
Exactly. You have PRC2, which is the enzyme that actively applies the duct tape, the repressive marks, and you have KDM6, which is the enzyme that removes them. They normally sit in a nice balance.
So in a WGD cell, is PRC2 hyperactive, or is KDM6 just failing to remove the tape?
It's the latter. The PRC2 complex is actually functioning at completely normal baseline levels.
Really?
Yeah, but the KDM6 enzymes are severely suppressed.
But why is KDM6 suddenly slacking off just because the cell doubled its genome?
To understand why, you have to look at what fuels KDM6. It is a metabolic enzyme that strictly requires a specific TCA cycle intermediate to function. It's called alpha-ketoglutarate.
Okay, alpha-ketoglutarate.
Right, but more importantly, KDM6 is highly sensitive to competitive inhibition by a different metabolite, succinate.
Oh, wow, this is fascinating because succinate and alpha-ketoglutarate are core components of the cell's mitochondrial energy cycle.
Yes.
So if there is a shift in that metabolic ratio,
Yeah.
specifically like a massive spike in succinate, it would outcompete the alpha-ketoglutarate, bind to KDM6, and just completely paralyze it.
Exactly. And when the researchers profiled the metabolomics of these WGD tumors, they found an abnormal massive accumulation of succinate.
Which makes sense because managing double the genetic material puts the cell under intense metabolic strain.
Immense stress.
It's the factory analogy. Imagine a factory that's suddenly forced into overdrive. It's churning out so much product to maintain this tetraploid state that it creates a massive toxic spill of its own byproduct, the succinate.
I like that.
And this succinate floods the factory floor, and it literally suffocates the KDM6 workers.
That perfectly mirrors the pathology. The team even identified that the WGD cells actively downregulate the subunits of succinate dehydrogenase, or SDH.
Which is the enzyme responsible for clearing the succinate.
Right. So the exhaust physically has nowhere to go. Because the tape removers, KDM6, are paralyzed by the succinate flood, the tape appliers, PRC2, are left entirely unopposed.
Oh.
They just run wild, applying that H3K27E3 methylation everywhere, packing the chromatin tighter and tighter until the alarm systems, the MHC-I genes, are totally silenced.
Okay, time for segment 3 recap. Point 1, WGD causes a massive metabolic shift, spiking succinate levels. Point 2, high succinate paralyzes the KDM6 tape remover enzyme. Point 3, without KDM6, PRC2 runs wild, accumulating H3K27E3 duct tape and silencing the immune genes.
It's an incredible cascade.
It really is. So listener, how wild is it that a simple metabolic traffic jam inside a cell's mitochondria can effectively shut down the body's entire immune response to a tumor?
It perfectly bridges genomics, metabolism, and immunology all into one pathway. And honestly, once they mapped that specific cascade, they finally had a blueprint for vulnerabilities.
Right, because if you know exactly how the factory is failing, you know exactly where to apply pressure to break it entirely, which moves us perfectly from the pathology into the therapeutics.
The best part.
Yeah, they didn't just map this, they ran a massive high-throughput drug screen against it.
They screened over 400 different small molecule compounds, and they tested them against both the normal diploid cancer cells and the WGD cancer cells.
Looking for synthetic lethality, right?
Exactly. Meaning a drug that the normal cells can easily tolerate, but that specifically destroys the WGD cells because of their unique stress biology.
And the biggest hit they found was a compound called YM155. Now, knowing the mechanism of YM155 makes this incredibly elegant.
It really does.
It targets a protein called BIRC5, which is widely known as survivin. Survivin is a potent anti-apoptotic protein, meaning it prevents the cell from initiating programmed cell death.
Which is absolutely critical for a WGD cell. Remember that running the engine past the red line creates massive proteotoxic stress.
They're unstable.
Deeply unstable. These tetraploid cells are constantly receiving internal signals to self-destruct because their genome is a total mess.
So they are perpetually teetering on the edge of the cliff of apoptosis. And they have to heavily upregulate survivin just to act as a crutch to keep themselves from falling over.
Right. While the normal diploid cells aren't under that kind of structural stress, so their reliance on survivin is super low.
So when they treated the models with YM155, they effectively just kicked out that crutch.
Exactly. The normal cells processed the stress and survived just fine, but the WGD cells, which were completely addicted to survive and to manage their genomic chaos, instantly triggered widespread apoptosis.
The tumors just collapsed.
It completely collapsed.
So YM155 exploits the structural and metabolic stress of the doubling event. But what about the invisibility cloak itself? Like, if we know the succinate buildup is paralyzing the demethylase, leaving PRC2 to hypermethylate the chromatin, couldn't we just target PRC2 directly?
That is the exact second major therapeutic angle they took.
Because if we block the enzyme applying the epigenetic tape, we should be able to force the chromatin open again.
Right. So they used a potent, highly specific PRC2 inhibitor called EED226. By introducing the small molecule, they chemically blocked the PRC2 complex.
So without PRC2 constantly applying the methyl groups, the epigenetic landscape starts to normalize.
And the in vivo results in the mice were just striking. Treating them with ED226 successfully erased the global H3K27E3 hypermethylation.
The duct tape dissolved.
It dissolved. As that repressive chromatin opened up, the cells regained access to the interferon gamma pathway. They immediately started transcribing B2M and MHC-I again.
They started manufacturing their ID badges again.
Yes. Antigen presentation was fully restored. And the very moment those cells became visible again, the CD8 positive T cells recognized the mutant peptides, flooded the tumor microenvironment, and initiated highly effective anti-tumor responses.
The WGD tumor shrank.
They shrank significantly.
We chemically stripped away the invisibility cloak. We forced the tumor to reveal itself so the native immune system could do what it's actually designed to do.
It's brilliant.
But here is the critical pivot. For anyone looking at actual clinical translation, this is all exquisite biology in a mouse model, sure. But does this precise metabolic epigenetic cascade hold up in actual human pathology?
The human clinical validation in this paper is arguably its strongest component. They didn't just stop at the mice, they queried massive human genomic and transcriptomic databases.
Like which ones?
They looked at the Metabric cohort, the TCGA database, and the FESCC cohort, representing thousands of human triple-negative breast cancer patients.
Okay, and when they stratified those patients by their whole genome doubling status, did they see the same immunological desert?
Down to the specific pathways. In human TNBC patients with WGD-positive tumors, they observed consistently lower overall immune infiltration scores.
Wow.
The tumors had significantly reduced expression of antigen presentation machinery, and they displayed the exact same enrichment for H3K27Me3 repressive marks that they mapped in the mice.
That is incredible consistency.
And it correlated directly with worse overall survival and higher rates of metastasis in the human patients.
They also utilized patient-derived organoids, or PDOs, right?
Yes, they did.
Which really bridges the gap between a database and a living patient. For the listeners, instead of growing flat 2D cell lines in a dish, you take a biopsy directly from a human patient and grow a miniature 3D version of their specific tumor in the lab.
Right, which maintains much of the complex microenvironment and metabolic architecture of the real tumor.
Exactly. So what did the organoids show?
They derived these organoids from human TNBC biopsies, segregated them by ploidy, and mapped their immune responses. And the WGD-positive human organoids actively blunted the activation and proliferation of co-cultured human T cells.
Validating the whole thing.
Completely confirming the active immunosuppression mechanism in living human tissue.
Okay, final recap for segment 4.
Yeah.
Point 1, we can exploit WGD vulnerabilities using YM155, which targets survivin to trigger apoptosis. Point 2, we can use ED226, a PRC2 inhibitor, to reverse the epigenetic silencing and reactivate the immune system. And point 3, this all holds true in massive human patient cohorts and 3D organoids.
Perfect.
So, listener, if you were an oncologist, would you rather attack the cancer cell's structural weakness by kicking out its crutch, or would you try to unmask it so the patient's own immune system can do the job?
It really opens up highly precise combination therapies where you could potentially do both.
Unmask the tumor first, then push it over the apoptotic edge. It's an incredible piece of cellular detective work. We track the pathology from a single misstep in cell division to a toxic accumulation of metabolic exhaust, right down to the physical condensation of the DNA around the immune pathways.
And more importantly, we see the tangible chemical interventions that can reverse it.
So what does this all mean?
Well, this raises an important question as we look toward the future of this research. If the entire immune evasion cascade is downstream of a purely metabolic shift, that buildup of succinate. Could we eventually intervene before the epigenetics are even altered?
Wait, really? Like, stop the duct tape before it's even applied?
Exactly. Could we modulate the tumor's metabolism directly? We might be entering an era where specific metabolic diets or systemic metabolic hacks that alter the alpha-ketoglutarate to succinate ratio could actually be deployed as a foundational form of immunotherapy.
Manipulating patient metabolism to literally change the way DNA is folded inside a cancer cell. That is a brilliant, mind-expanding concept to leave you with.
It really makes you rethink everything.
It does. Thank you for joining us as we took this deep dive into the microscopic battleground of whole genome double breast cancers. It's a powerful reminder that the mechanisms cancer uses to hide are often the exact vulnerabilities we can exploit to destroy them. Keep asking questions, stay insanely curious, and we'll see you on the next deep dive.